Good morning. Thank you, everyone, for coming here and celebrating with us and having this experience with Vish coming and talking about what he's been doing for the past couple of years. It's been an adventure. I'm talking to people online mostly, and I don't know how many people are out there, but trying out here. What was it, 5 years ago? You came in one fall just saying, hey, I'd like to do a little bit of research, not really thinking about a master's, but we'll try it out. And as 3 weeks later, 4 weeks later, it was like, I can't let you go. I got you on an RA and got you into the program. You didn't know you were a PhD student yet, but the rest of us did. And somehow we convinced you to stick around and got you in here. Managed to survive the whole master's graduation process with a lot of trepidation on my part, not yours. I was worried about, are we going to lose Vish? You know, he'd be really great. And here we are a couple of years later, and it still seems like the very first semester, just the amount of energy, the enthusiasm, the tackling of the projects. And every day we discover something, we learn something. Something is interesting and curious. It's broken, but something that we can move forward. And I've just truly and deeply appreciated how you've brought that to our lab. And I'm excited to see how you've put all of this together, and we're going to share it with the community and sort of see how all of this goes. So thank you very much. The stage is yours.
You don't want to graduate, right?
Oh, I know, yeah. It would be fantastic if you decided to stick around for another 5 years.
That was a puzzle looking for it. Got it.
Well, fail me if you will. I'd love to be here. Thank you for.
Wait, is Holger on?
Yes, he's on. Yes.
And I would like to make a secondary special thanks for the connection with Holger Roth up at NVIDIA, who's become a good friend in all of this and a great collaborator, really a co-mentor with Vish. It's been a lot of fun to see how these projects have come together. It's unfortunate you can't be here now, but we have a plane ticket. And as soon as we have things normalized, you have an open invitation to come on down, and we'd love to hear from you, give you a tour of Nashville, and get our collaborations going even more deeply, because hopefully Vish will be up there. Vish will be up there in a little while, and sort of keep all of this thing snowballing. So thank you so much, Holger. It's been a pleasure, and I hope this is just the very first start of working together and keeping all things rolling.
That was a very genuine introduction. Thank you. I loved it, Dr. Lemon. I'll start with the title of my thesis. It's called Integrating Histology and Microarchitecture Modeling with Deep Learning for Diffusion-Weighted Magnetic Resonance. But those are a lot of words, and even I did not understand any of them when I was beginning out on this fulfilling journey of scientific exploration. Just like Dr. Lannon mentioned, I was looking for a little research. However, I think it is my predicament to be able to explain some of these to an extent. And to the general audience, in short, what we're going to do is we're going to use MRI. MRI is familiar terminology, right? To look at tissue and estimate histology, and we're going to make it more specific. I'm going to make it more reproducible, make the estimate of microstructure closer to histology, and that's what we're going to look at today. So before we begin, I've divided my talk into 3 different sections. The first is where I'm covering the fundamental components, where I've got the titles for most of the keywords that I said in this huge long title. The second phase is the contributions, which is my scientific journey. And then phase 3, there are some concluding notes and applications and a little bit of future work. All right, why do we even care about diffusion MRI? Diffusion-weighted magnetic resonance imaging is being used worldwide to produce images of brain tissue structure to characterize the diffusion process at a cellular level to generate contrast for brain images. And it has emerged as a promising modality for studying brain connectivity for both normal and the diseased brain. It has opened new lines of inquiry for human neuroscience and for understanding of brain illnesses. This modality holds a lot of potential, and it can help out hundreds, thousands, or even millions of people with all of these kinds of diseases. Currently, Diffusion is being used for clinical diagnosis of stroke. It's being used to study neurological disorders such as multiple sclerosis. A new upcoming application has been detection of cancer, metastasis. One can also detect segmentation of the white matter structure of the brain. And then we have the microstructure or microarchitecture, which are estimated from neuroimaging mostly. And the number of publications with microstructure and diffusion have doubled like a clockwork every 3 years since 1995. And the reason is because this modality holds the potential to help out with so many diseases for all the people who are suffering from them. So what are we actually measuring with diffusion MRI? If we look at this equation here, that measurement S, which is acquired as a diffusion-weighted signal measurement, is actually a signal attenuation, which is representative of random water motion for white matter tissue of the human in vivo brain. Now, the cool part here is that we do not just acquire one single volume. We acquire lots of volumes, and the number of these volumes corresponds to the number of points spread on this hemisphere. And so what we're actually looking at, we're looking at a specific gradient volume in a 3D direction. And that's what this is. And here's another one, which is in another direction. And just as an example, the signal attenuation difference can be noticed here with the colored blue and red arrows, which are pointing out regions of signal attenuation in the corpus callosum towards the left and the right. There is also another important scanning acquisition parameter, which is called the diffusivity value, and it measures the intensity of diffusion weighting. This diffusion weighting affects the contrasts of these images. This effect is observable for the different diffusivity values of 1,000, 2,000 seconds per millimeter square, and 3,000, and we can see the change in the contrast. And all these three volumes are scanned in different directions with a different diffusion weighting. So now we have this crazy kind of an acquisition. What do we do with this? Well, we model. And trust me, you can do a lot of different kinds of model with diffusion MRI. For now, I'll just show a couple of them. But if we took this equation and I said, I'm going to block out this mono exponential model. And I said, you can do whatever you like with this. This is your magic box. Because modeling diffusion-weighted MRI is an inverse problem. Because the measurement that we get out of the scanner is the white S. And we are free to model it in any way we want. And you can fit in that red box. Now, of course, it's not whatever. This is going to be within the scientific boundaries. Here, we're looking at the corpus callosum again. And we have a signal representation model. And it's the same equation, just that it has been modeled with a signal model where C represents coefficients and Y is the basis for representation. That said, the geometric structure of the signal shows us attenuation in the horizontal plane. You could almost pass a line through it like that. And the second one is a tissue orientation model. If you take a look at this model, then the tissue structure is flowing right through the signal. And so that is how like this tissue geometry corresponds to the signal, because where the signal attenuates, the tissue exists. And again, we have the same equation, just that it has been modeled in a different way. Now I'll give an overview of like a bird's eye kind of view on diffusion MRI. We start with the human brain, we take MR images, you do pre-processing, basically to make the data better. And then you can do microstructure methods like the tissue orientation models, which I just showed. And then you can construct roadmaps of the brain, which is called tractography. That is essentially the white matter structural connectivity. So how did all of this modeling begin? It began with diffusion tensor imaging. Diffusion tensor imaging was proposed in 1994. However, it was limited. And the primary limitation was that can only detect a single fiber orientation. And you can see that if you look at the fanning fiber case, the green ellipsoid and versus the crossing fiber population, it does not change, it is the same. And so what we needed were advanced methods. These advanced methods are called Hardy methods. It stands for high angular resolution diffusion imaging. For simplicity, we'll just call it Hardy. They're advanced methods that can detect multiple fiber crossings. Now, Hardy was not a single method. There are lots of them. A popular one is super resolved constraint spherical deconvolution. We'll call it CSD because we're going to use it a lot. It's a classical transformation and it has been used since the last 15 years. There are lots of them: cue ball, diffusion orientation, persistent angular structure, shard, multi-tissue CSD, forecast, naughty, and even challenge studies have been conducted which show a comparison of all of these different methods. And what they defined was that in order to say that it is Hardy, you can go define it by the acquisition. If I have more than 45 gradient volumes in different directions, then I can call it a Hardy acquisition, and these kind of methods can be fitted. Now, all of these methods are affected by tractography directly. I'll show an example. So over here, we're looking at the frontal lobe in the human brain, and we have two different kinds of CSD implemented on which tractography was done. And so if you look at the top versus the bottom, you can see that the bottom has more noisy and spurious tracts, like in the zoomed version right here. And what this tells us that either we need an advanced method to improve tract reconstruction or a higher quality acquisition. So what is it limiting? What is it that is limiting us then? Well, understanding the limitations for a field requires covering a huge amount of literature, and every published paper has its own story to tell. I'm only going to show a small fraction of what I covered to give a brief overview. This one right here is a specificity study where phantoms were used to evaluate the specificity of cue ball. However, phantom data sets cannot be equated for a human in vivo data. There are method comparison studies on synthetic data sets, and all of these colored plots are different methods. Then tractography studies have been done on synthetic brains. Histology-based validation studies have been performed where the ground truth is coming from histology and the comparison is being performed through MRI. Now, these studies have really few voxels. The same story is repeated on a rat brain. Then we have another one on human postmortem tissue. And the catch is that the methods really do not agree with all the prediction, with the ground truth of histology. And then here's another problem where if we take one population of subjects, and if we scan them on two different scanning sites, just think of it scanner A, scanner B, and if we plotted out a metric, then we can see there's a shift there between the gray versus the red. And that is a bias coming from the site. So we know there's a problem that needs to be harmonized. Here's another example of the same. So we know now that there are lots of problems. Summarizing all of them together, the empirical characterization is limited. We either have phantom studies or small-scale data sets. Then the microstructure methods, they're limited by classical transforms and the lack of 3D ground truth data sets. And then harmonization, where the scalar metrics are inconsistent with each other, and these metrics should actually be acquisition independent. Now, we knew that looking at all of these gaps, we needed an integration for the two modalities of histology and MRI. But we did not know how either. However, data-driven models were coming up in a big way, and specifically deep learning was showing a lot of promising results. So where did deep learning come from? And what does it do, and how does it work? Deep learning is inspired from non-learning models for neuronal brain activity in 1943. They were proposed by McCulloch and Pitts. Somewhere a decade later, Rosenblatt said that we can get these neuronal models to actually learn. And then it took us 4 decades to actually get the hardware to compute all of these learning models. And now we have lots and lots of architectures which are learning different things for different problems. And it's being applied to a lot of different fields. We've got genomics, we've got natural language processing, and then of course we have medical imaging. What can this deep learning actually learn? A simplest example, this is a computer vision task. If I have an image of a cat versus a dog, a deep learning network can classify whether it's a cat or a dog given that image, if it was able to learn. Here's another advanced example where, given this image, it detects that there is a group of people and that there are many vegetables on the fruit stand. And then it can also detect complex things such as the highlighted objects in the image, for example, the frisbee, a dog, a stop sign. So how does this deep learning model actually learn? Okay, don't worry about the math. I'm just gonna keep this abstract and really simple. So we have a 2-layer network because the bottom row is the input and the top row is the output. And right here, the middle 2 rows, these are our learning parameters. So the input can be anything such as images or text. And then it can make predictions in terms of classification probabilities, for example, just relating it with the cat or the dog. And so what actually happens is that when we propagate forward, the input given gets transformed through these learning parameters, and it eventually gets converted by feature extraction to the classification probabilities. And we have a prediction now. So once we have this prediction, we can compute error. Error is a simple term which is taken between the ground truth and versus the prediction. Now, if we took a small fraction that you can imagine of this error, and we sent it back to update the parameters, the learning parameters, then this causes learning. And that effect, if this is repeated thousands and thousands of times, then this deep learning model is actually able to learn. Deep learning and diffusion-weighted MRI were not that simple. They had their own specific set of challenges. If medical imaging data sets were 2-dimensional, it would be great. Unfortunately, it's not so, which is why we are all here today. Firstly, the data is a 3D volume, which is why we're dealing with voxels instead of pixels. One could think of them like 3-dimensional pixels. Secondly, we have many gradient volumes, which makes this data 4-dimensional. And then if there are multiple shells, and if they vary, then this data becomes 5-dimensional. And so these are the challenges, which was pretty hard. And these were all open-ended problems that we did not know the answer to, of how to drive these powerful deep learning models to learn useful information from diffusion-weighted MRI. Now, this is the end of the first phase of my talk. And we'll be going through the second phase where I'm going to talk about the things that we learned along the way. So far, we've covered the basics of diffusion MRI and why it's important, an overview of microarchitecture modeling. And we know that there are lots of problems. We knew that the empirical characterization was limited because of phantom studies or synthetic data sets. So what we're going to do is we're going to characterize empirical reproducibility on one of the largest single subject data sets ever. This data was acquired with 5 different diffusivity shells and 96 directions per shell, and that was repeated 11 times. The total was over 5,800 volumes. This data was collected over 3 days, and the total scanning time was over 6 hours. Now, empirical characterization for human data does not have a ground truth for the simple reason you can't simply access the brain. You can't. So we had to come up with a hypothesis of how we were going to study this. So since we had a lot of data, we could construct a gold standard by concatenating all the data together per diffusivity value. And our idea was to compare a single scan versus the gold standard across different diffusivity values like that. We took two different Hardy methods of pass and cue ball, and we can see these visually in the left side frontal lobe of white matter. We did an intra and inter-method comparison, which means that we compared pass gold standard versus pass on a single scan. Similarly, cue ball gold standard versus cue ball single scan. And inter would be exchanging between the two methods. And knowing that there were problems, we did not have any high expectation. We thought that these methods would be moderately consistent since we knew there were problems even with small scale data sets. However, if I compared them intra-method, which is where I say that I'm just looking at the number of fiber orientations of the gold versus the single, then the number of fiber populations are quite similar. I'm just counting the number of fibers and that's it. It's just counting. But if I compared them inter-method, then the agreement is hardly there. If just for the single fiber, like it's all white, there's nothing there. If it was red, that means high agreement. And for the two fiber, even if agreement is there, it's really, really low. This was totally unexpected. We still plowed on. We wanted to see if the tracks were doing any better. And so for that, we hosted an international challenge with a lot of data. We had a single subject that was who scanned 5 times for 2 sessions per scanner on 2 different scanners. The total number of radiant volumes was 2,640. And this international challenge focused on assessing reproducibility of tractography algorithms for 16 different tracks. And 9 international teams made a total of 45 submissions to this challenge. Let's take a look at what happened. If I put all of those algorithms on each other, it does not take a neuroscientist to figure out that these tracts cannot possibly cover the entire brain or even half of the brain. What it should have looked like was kind of this. Again, the variance was extremely high. Summarizing this entire story, the microstructure methods, if I intra-compared them, which is just in itself, then it's great. But among the different methods, it's very limited agreement. The tractography challenge shows that there is an extremely high variance for all the tracts. And that led us to publishing a lot of work in awesome journals and top-tier conferences. And the international challenge is still being held live, and people can still participate and put in their tractography algorithm for assessment. We just saw how inconsistent these microstructure modeling methods were. The tracts were not telling a different story either. And this was the point in my story of research where I needed to make microstructure estimation more consistent and reproducible. So guess what? We're going to take an extremely rare squirrel monkey histology data set and see if we can even learn a little relevant microstructure. There's fantastic work behind the construction of this data set from Dr. Schilling, Dr. Gao, and Dr. Anderson, who's sitting right here. The data set took over 10 years in formation itself and holds multiple PhD dissertations behind it. The construction comprises of scanning 3 ex vivo MRI squirrel monkeys, 3 squirrel monkeys with ex vivo MRI scans, after which histology sectioning was performed and stepwise registration procedures were put in place to put MRI and histology to correspond with each other. However, the sample size of this data was still extremely limited. We had only a total of 567 voxels from 3 monkeys, which is an extremely small data set for learning models. Our chances were slim of learning on this data, but we had to try. Well, we tried out this deep learning model, which is using 5 hidden layers, just like the previous deep learning I showed with 2. This one is using 5. The input is based on the signal model that I've shown earlier. And then we have the ground truth coming from histology-based microstructure. And so we utilized the training data of just 2 monkeys, and we left out the third for testing. But before I get to the results of the third monkey, I have something really cool to show, because we learned on this model and then we made predictions on a human brain. And so this is going to sound really weird, but we learned from a monkey. And if you look at the red box over there, those are predictions versus what is coming out of a classical method, which is right here below, then the geometric structure is flowing in a visually consistent way. And that was crazy for us. We learned on a monkey and predicted on a human. I still can't believe it, but that's what we did. Now, things were getting interesting. We needed some kind of a metric that could make sense qualitatively and quantitatively for comparison. And so this metric has been proposed in literature before. It's called angular correlation. It just measures how similar things are on a sphere, and that's it. And I'll give a visual understanding of this metric as well, because we want to make it sense of it quantitatively and qualitatively both. This correlation is measured on a scale of -1 to 1, where 1 is like good correlation. It means great. So over here, we're looking at a single subject, and the data is from scanner A and scanner B. So it's 2 scans. Now, the underlay over here behind the geometric structure is the correlation, and that is the scalar measure. So if we looked at the positive correlation regions, look at that box. If you compare the two, the structure is quite similar, and the ACC is all bright and lit up, which means that it corresponds qualitatively and quantitatively. And if we looked at the negative correlation regions, then the structure does not agree. And the ACC is also pretty dark and gloomy behind. Now that we know of a good measure that makes sense, I'll show the results on what happened with the third monkey. We compared on a lot of different methods. And just for simplicity, I'll highlight the proposed method of deep learning. This correlation was the highest when compared against all these other 7 popular methods that are out there. We also took this same method and tested it on human data by testing it on scan and rescan acquisitions on 12 different human subjects. And if we look at the yellow curve, that is the proposed model, it shows the highest correlation as compared to the other 2, which are traditional models. Well known, they are variations of CSD. And if we looked at independent distributions, then we can also see that the deep learning model shows the highest correlation, which is at the top, the rightmost plot. Let's see now. We've covered problems. We covered empirical characterization. We can learn off on monkeys, predict on humans. Sounds a bit weird. But we wanted to ask the questions, could we harmonize across different scanners? And what that led us to is this is same way, single subject on scanner A and B. And we were asking these questions because we found that the correlation was low. It was getting better with these data-driven models, but could we really build something even better? And then it wasn't possible to intrinsically harmonize. And what that means is that it should be indifferent to any kind of scanner site. It should not be specific to a scanner site. If these models could learn on animal data sets, could I integrate it with the human data? And so we came up with the hypothesis to do it. And how we did it, before we get to how we did it, was I'm gonna show what the data looks like. So I'm gonna go slowly on this one because there's a lot going on with this slide. So we had we had 3 subjects. Subject 1 was scanned on site A and B. Subject 2 again on A and B. And the 3rd subject was scanned on A and C. And I know the C is highlighted in red, but I'll get to it. There's a reason why. And all of these are human in vivo acquisitions on a single shell of data. And what we wanted to do was integrate this animal histology data set with a single subject. So for training, we just used subject 1 on A and B and the histology data. For testing, we used the data from subject 2 and 3, and that is why I've highlighted C in red, because that scanner is totally unseen to the training data, because we're only gonna use data from site A and B. We're back to the deep learning model over here. And the way to incorporate human in vivo data was that if we took a corresponding pair of voxels, one voxel coming from site A and the other voxel coming from site B, then we could pass it through the network, but we didn't know what the output should be because we don't know what the ground truth is. But we know that the differences should be 0. And so what we hypothesized here was that the squared sum of the predictions for these two should be 0. And so we incorporated that in our own error term. And what that led us to was these results. And so if we looked at the correlation for the subject 3, where we're comparing between site A and site C, we've got the highest correlation as compared to the other two. The green line is the prior data-driven model. The red one is a traditional model. And this story is repeated for subject 2. But again, the data was seen by the model because that's from site A and B. Looking at all of this visually, we can look here. The correlation is pretty dark and gloomy there. If we go to a data-driven model, it improves. And as we get to the new null space model, then it improves even more. And that led us to proposing a lot of work and publishing in top-tier conferences such as International Society of MRM, MICAI, and SPIE. And that's the end of the 3rd contribution on harmonization of microstructure. We showed problems we learned on monkeys. We can intrinsically harmonize now, but we have not even entered the 5th dimensionality of the data-driven modeling yet, which is the multi-shell data. So if you look at this figure, at the top row, that's a single shell acquisition, and we have the geometric structure coming out of there. We have traditional methods and we have data-driven methods. Even though these models were barely scratching the surface, but they're working pretty well. However, no methods existed for multi-shell diffusion modeling for if we talked in terms of data-driven modeling. And so I'm gonna give a brief recap of this data because I wanted to mention that this data set of histology actually had multiple shells of diffusivity values. It had B values of 3,000, 6,000, 9, and 12,000. And each had 100 gradient directions. And so how we actually learned on this, I'm gonna take a small recap to the signal model. Now, if you see a lot of math equations, please don't worry. We're only talking about these in an abstract form of way. We modeled a single shell signal model like this, and those set of coefficients can model all of these geometric structure for the signal. However, if we take multi-shell data, then if I wanted to model all 4 shells together, these coefficients cannot be captured. And the reason for that is because the basis is just angular. And what an angular means is because the gradient directions are spread over the sphere, so it's only angular. There is no radial component there. In order to model that, we had to introduce another radial scaling basis there, which could capture all of these together. And so in order to be able to learn between the diffusion MRI and microstructure, we use the combination of angular and radial space for microstructure, ground truth of histology, and MRI both. So the results that I'm showing here, I'm always predicting on all 4 shells of data given any combination for learning. And what I mean by any combination of learning, whether I learned on a single shell of data, which would be the top left, and then the top right is learning on 2 shells, and then the bottom we have 3 and 4 similarly. We could always make better predictions as compared to learning just from an angular basis, as compared to where we are utilizing both angular and radial basis for learning. And we can look at this visually as well on a human in vivo subject. We're looking again at the left side frontal lobe of white matter, and the correlation here is the highest. That was a bit complicated even for me. So a little recap, we've looked at empirical reproducibility, we have data-driven methods, we could harmonize, we can learn on multi-shell data, and that led us to a very interesting question. We wanted to know if it was possible to recover multi-shell information from just a single shell of diffusion MRI. And let me give an overview of why we were even asking this question. So multi-shell acquisitions allow us to model advanced models like MTCSD, and that leads us to high definition fiber tractography, which is actually quite relevant for pre-neurosurgical guidance information. And we wanted to know if there was a way where we could use just single shell information and get to high definition fiber tractography. So our hypothesis here was, if we had a network structure and we have 4 shells of information, 3 shells of information, and we can construct a ground truth of CSD, then was it possible to just use a single shell and incorporate the neighboring information that a voxel has, which would be, if we were looking at a single cubic voxel, now we're looking at multiple voxels. So this represents a total of 27 voxels because it's a cube of 3 by 3 by 3. For this hypothesis, we used only human in vivo data because we're only going from single shell to multiple shell. We use data from the Human Connectome project, which has multi-shell acquisitions. And we had a total of 15 subjects. We used 5 for training, 2 for validation, and 8 for testing. However, that should not lead you to think that the sample sizes was low, as a single volume of DW-MRI has more than 680,000 brain voxels, and that translates to over 2.5 million patches. So there are a lot of voxels for training. And if we took a look at the results, then just the traditional single shell method, which is the blue plot there, has very low correlation with multi-shell information tissue orientation. If we look at the prior data-driven model, which is the green plot there, then it has a bit higher, but not as high as the red one, which can be achieved by incorporating spatial information. And so we also tested the distributions on individual subjects. So the distribution gets skewed towards higher correlation like that. We can see it getting constricted if you look at this. And so I promised you tractography, and so that's what I'm actually going to show. On the right-hand side, we have the ground truth of tractography coming from a very high quality acquisition. We're looking at the corpus callosum tract. It connects the 2 hemispheres of the brain, the left and the right. And if you look at this region of occipital lobe here, then you can see that there are a lot of branches. And in the earlier method, if you look there are not that many branches. But with this proposed method, we can come up with a quite more branching relatively as compared to the method which is showing the red box. And a similar observation can be made here at the top in the same way. And I'm also going to show the arcuate tract. This tract actually connects your vocal cords to the brain. And again, this is much easier to see, and it's pretty much self-explanatory. But over here, there's a lot of branching. There's no branching here at all. Over here, it's much better as compared to the red box. These contributions together, again, led us to publishing a lot of work at top-tier conferences for SPI and MICAI and ISMRM. And these are the end of my scientific contributions. And now I'm just going to briefly show you a data-driven, an application based on these data-driven models before we get to the conclusion. And so here I'm presenting a data, and this is a 56-year-old male, and he was presented with asymptomatic right-sided temporal occipital lesion. It's evident right there in the contrast. And this acquisition, it has one shell and only 30 gradient directions. And 30 gradient directions is not even hardy because it's not 45. So this is drastically less. But if we made predictions, firstly, the tissue orientation can actually capture this lesion. And if we perform tractography, we can see that high resolution tracts can be recovered here. We can see red is the arcuate, the yellow is the corticospinal tract, and there are many others. What it looks like when actually compared with the traditional approach. So if we look for the corticospinal tract, then on the left side top, there are way lesser streamlines as compared to the right one. And a similar observation can be made in the below plot. It's just that it's been presented in a different view. And the story repeats itself for the singular tract and the frontal aslant tract. And with that, I'm gonna conclude that we've barely scratched the surface for data-driven modeling, but they're working pretty well given for how much data we had with histology and with the incorporation of spatial information. However, there's lots more that needs to be done. We proposed methods that could intrinsically harmonize and can generalize to unseen scanner sites. And then we showed recovery of multi-shell information to a really good extent as compared to information which is just coming from a single shell of information. And with that, I'd like to conclude and thank you, everybody. First, I would like to thank my lab, and then I will always be grateful, and even that is an understatement. Thank you.
You ask the audience, so we'll have public questions for a while.
Yeah.
I will read them off here, or if you can't see them, so